Hi, I'm Ted Clancy, and this is Eye on WP. Hello, my name is Ted Clancy, and I'm your host on this edition of Eye on WP. We have many excellent programs here at the university, and today we'd like to cast our eye on one that has been a strong contributor on campus for many decades. It's a department that has developed in the technology age, but still prides itself with a vast amount of hands-on, roll-up-your-sleeves, and don-your-hip-waders kinds of teaching and student involvement. Look down any hallway at William Paterson University, and the modest appearance belies the excitement and adventure that lies behind its doors. In line with the university's commitment to student success, we'll find students doing hands-on work with chemicals, modern equipment, and advanced technology. But students, even undergraduate students, have significant opportunities to work with live animals in clinical as well as in field studies. The students have a chance to get their feet wet with field experience and use technology to further understand family and group dynamics of other species. Students get a grasp on the inner life of plants through studies of plant genetics. Come with us as we open the door and explore the exciting world of William Paterson University's biology department. Recently, I met with several biology department professors to discuss with them what kinds of work they were involved in, how it helped the students, and indeed how they hoped it would help humanity. The first professor I met with was Dr. Marty Hahn, and I asked him about his work at William Paterson and what motivates him.
That has to be, the bottom line has to be curiosity. I remember in school I was thinking about being a clinical psychologist, so I took several courses. I was all ready to go, and then I took a course in animal behavior, and I was just fascinated by watching animals, seeing what they're doing, and asking myself some very seemingly simplistic questions like, gee, I wonder if mice can tell each other apart. And as it turns out, I'm still working on that 30 years later. So I've been interested in mouse social behavior. In particular, I'm interested in the interactions between infant mice and their mother. And it turns out that infant mice produce a number of sounds, several of which have signal value. That is, they make a call or a sound to mother, and mother responds in some way. So you get into mouse communication. The infant signals, and mom does something about that signal. So that, in particular, mice produce ultrasonic calls, that is, calls around 70 kilohertz. That's about 3 times as high as our highest hearing can go, so it's a very high-pitched sound. We can't hear the sounds. We have to use special instruments in order to be able to know that they're producing these sounds. And they make these sounds early on, right after they're born, and they stop producing them, infant mice stop producing them about 12 or 13 days. There are not that many institutions in the country where undergraduates get to work with experimental animals. That it's a privilege that they get, and I think they recognize that when they walk in. That's why I try to be a good custodian, and Norm does too, of the welfare of our animals. We take that very seriously. Well, Norm is really the heart and soul of the laboratory. I could use many colloquialisms here to apply. He's my right hand. He's the guy who's here all the time. He tells me what's going on. So he comes up here in the middle of the night to do something if it has to be done. He's here on the weekends to make sure everything's okay. He's integrated into the mouse lab. I described us as a family. I'm not going to pick a family member, but certainly Norm is the mainstay of the lab.
This is my 16th year here. I do have a lot of collaboration with the professors in their research, where I do have several papers with my name on it. So we do work very well together in that sense.
And we do some work, although my heart really is in understanding the mouse as a mouse. We do some work in which the mouse is a substitute for humans. If you're interested in mouse anxiety, as some of my colleagues are, then you can introduce situations that'll make a mouse more anxious or less anxious, and you can monitor their behaviors. You can even use drugs which increase anxiety or decrease anxiety, and so you can study the mouse as a model of human anxiety, something which is very important and that a lot of investigators around the country, around the world, are working on now.
One of Marty's longtime colleagues is Professor Bob Benno. Bob and I had a chance to discuss many serious aspects of his work, as well as some of the more humorous ones.
It's interesting. Whenever I tell people that, this is what I do, I study prenatal drugs, everybody always, oh yeah, but it's such an important aspect of our society, and to really look at the long-term consequences of drugs upon human behavior. So. And I think that's why I wound up going into the neuroscience, was just to try to find out more about drugs and behavior and how we function. And we focused specifically on, for years, on cocaine, and more recently the effects of cocaine and alcohol upon, prenatal cocaine and alcohol upon development and long-term behavioral changes in offspring exposed to those things. Interestingly, a lot of this work with the prenatal cocaine has shown that by using our animal studies, that there's not as great of effect upon offspring with prenatal cocaine as one would be led to expect. And one of the things that we've found over the years is that there are small but reproducible changes in behavior, but it's not as strong as one would suggest from the literature in humans and the crack syndrome. And over the years, we've grown to realize that probably there's something more involved, that the humans who are exposed to crack also are probably exposed to alcohol and are malnourished. So a lot of the problems that are associated with that probably involve something more, a multi-drug abuse and poor prenatal care. Some of the minimal changes we did find seem to be possible subtle changes with behavioral set, which we took to interpret that there are an inability to respond appropriately to complex behavioral situations. And while that's not like, prenatal alcohol, where you have somebody born with a low IQ and all sorts of morphological changes and changes in fetal features and such, they are important changes for long-term consequences of human behavior. So if you can't make a proper decision what school to go to, or what job to take, or what town to move to, or what to do in a situation that requires you being able to make some, very subtle behavioral decisions, you can run into problems. So that was one of the major findings from our work.
Addictions of all types are so prevalent in our society, and it's gratifying to me to see so much exciting research being done to try to understand the complex nature of these behaviors. Another faculty member whose work in many ways complements the work of Professors Benno and Hahn is Dr. Emmanuel Onivy. He took a moment to explain to me the nature of his research.
The big area of our research is on drugs of abuse, including alcohol, how it affects the brain and behavior, and also how to be able to scale this down from our animal models all the way to the human behavior and function. So we are looking right now at this new system that has been discovered in the human body that we didn't know that existed before. And the new finding, the new explosion, and the new excitement about this is the discovery that the human body itself makes its own marijuana-like substances that are referred to as endocannabinoids. That means marijuana-like substances produced by the brain, the testes, the uterus, and all parts of the human body. And if the human body produces this, there must be genes that code for the proteins that are affected by this. This is really very simple in terms of being able to involve students to conduct research on this aspect. And this system of marijuana in the human body and brain is the most abundant chemistry in the human body and brain and testes. This is completely mind-blowing.
Who'd ever suspect that the human body would or could produce these kinds of substances? Dr. Onaivi went on to explain that the newly discovered endocannabinoid system interacts with previously known systems in the human body. This has led to their current research involving the relationships between personal tastes for hot, spicy food and marijuana receptors.
The very exciting research that we are doing now is to understand how the interaction between these marijuana-like systems that the human body have and other systems that are known. For example, we have concentrated and recognized for this the interaction between capsaicin and cannabinoids. And perhaps this might form a basis to understand why some people like hot peppers and some people don't like hot peppers. So this is an area of focus that we are using animal models like the plus maze test, the depression model, the condition place preference, the two-compartment black and white box, and the activity meters to be able to understand how these affect both behavior and the biology.
Some of Dr. Onaivi's students were recognized at the national level for some of their contributions in this area.
We're very excited and happy that there's an association in the United States called CORE, the Curriculum on Undergraduate Institutions. This society gets together and select competitively research from undergraduate institutions in America. And they choose about 50 to 60 students to showcase undergraduate research before the Congress. And the students make a poster. Our students from William Paterson University were competitively selected to represent New Jersey from amongst the institutions in New Jersey, and we're not the big school. So that was very humbling for us and very exciting for us that we are selected to represent New Jersey and present student research before the Congress of the United States. We are also very honored and humbled that the Board of Trustees of the university recognized this, because this had never been done before in the university, to be recognized and to represent the state. So we're very happy about that.
It's wonderful that Dr. Onaivi and his students have been able to garner such acclaim for themselves and for William Paterson in the short time that he has been here. I asked him what made him decide to come to William Paterson University.
William Paterson is a small university compared to the big research institutions, and therefore you guys can help me here, you can be a big fish
in a small pond.
In a small pond, you see. And in that situation, it makes a lot of sense, as you can see from the progress in the 4 years that I've been here, that we've continued to make in terms of research and teaching students and service to the community, that is being recognized nationally, locally here, and internationally. So this is very exciting for us. This is very exciting for me in particular in terms of this new vision and this new energy that we bring to the system.
When we return, we'll migrate from the animal world to the botanical with Professor David Slaymaker.
Push-ups, and she's 85 years old!
For more info, go to OperationGraduation.com.
Welcome back to Eye on WP as we continue to explore the fascinating worlds behind the modest doors of the biology department at William Paterson University. Professor David Slaymaker and his students work with plants, examining their natural defense systems and how to improve on them to foster greater crop yields. This has implications for many facets of human life.
What my lab works on right now is how plants defend themselves against pathogens. So there are different bacteria, fungi, and viruses that cause diseases on plants. And of course, for growers who are growing plants for food particularly, these diseases cause a lot of losses. So we want to understand how plants naturally are able to protect themselves from these pathogens, which in many cases they can. So my lab looks at how these plants accomplish that. And what we do, and this is myself and various undergraduate students, right now I have 2 undergraduates working in the laboratory. What we do is we look at how plants are responding when attacked with or exposed to a certain pathogen, and then we manipulate that response to look at how important that response really is in the success of the plant, and also whether that response can be manipulated really at the genetic level in order to improve the ability of plants to resist diseases in the field. And of course, this is with the hope of producing a more consistent and reliable food supply, and also allowing growers not to have to apply so many chemical pesticides, which are necessary in many cases today.
Professor Slaymaker's research also involves manipulating DNA. Working with DNA must be very interesting and difficult. Obviously, you can't see it or touch it.
Yeah, working with DNA is a challenge. It's really a conceptual challenge because working with DNA, like you said, you don't see the DNA. It's not like working with the whole plant. With the whole plant, you see the leaves, the stems, the roots. It's right there in front of you. So DNA works much more abstract, which is really good for students because it challenges them to apply what they've learned in classes to an applied method, procedure, and experiment. So they have to think about it, to understand it, in order to be able to know what to do with it, how to test it, and how to interpret those results as whether did this really work, did it not, and if not, why not, what do I have to do to make it right.
Professor Slaymaker explained to me that the manipulation of DNA is only one aspect of their research. After all of this DNA work has been done, what happens next? Where does it go?
The tissue culture facility that we'll see contains two different chambers. The first is the tissue culture chamber. It's specifically made in order to provide a very low light and the right temperature conditions in order to allow these tissues to develop into stems and leaves and roots. And in that chamber, we'll see two different types of containers. We'll see some small flat plates where we take that piece of leaf tissue and put it onto a medium, and that medium provides hormones and minerals and nutrients required for those specific cells that took up that DNA in order to be kind of induced to make a stem, to make leaves. And actually on those small plates, we only make stems and leaves. At that point, we remove those shoots and put them into these little magenta boxes. They're called small boxes, again with media in the bottom, solid media. Poke that shoot into that media, and that media has hormones and nutrients that induce that shoot to now make roots. And that's where we get the entire plant. So once we have decent-sized plantlets in those boxes, then we move to the second chamber, which you'll also see. The second chamber is actually kind of my baby. It's brand new. We just got it this year. And it was purchased actually through an incentive grant from the provost's office in order to expand the research opportunities for students and the interaction of students and faculty and research. And what this chamber does is this chamber has stronger light conditions. It has more space because the plants are going to now be growing to almost full size. And we grow them covered in an enclosed environment where they first adapt to light and to the soil, and then we open up that enclosed chamber. And now, of course, they're exposed to the low humidity of the air and also to the bacteria and fungi that float around naturally in the air. So this chamber is like an intermediate step. So what we'll see in that chamber right now are plants that have just been opened up. So you'll still see the plastic wrap around the lid of the pots and very small plants, and some plants that were opened up about 2 weeks ago and which are almost ready to go to the greenhouse.
After seeing the various chambers, David and I visited the William Paterson greenhouses to experience the next phase of the transgenic plant process.
So in here we've got the greenhouse. This is the final stage of the plant process, of the transgenic plant process. We can do a couple different experiments, including some bacterial inoculations in the greenhouse. We can also look at morphological characteristics. And the reason some of these have bags over them is because they're starting to flower. And we want each of these individual plants to carry on its own pure genetic line. So this way they don't breed between themselves, and each of these plants we've created maintains its genetic purity from generation to generation. It gives us reproducible study subjects over the years.
We then left the tobacco plants and visited the second greenhouse, which houses a highly varied cross-section of plant life.
So here we've got a diversity of different plants that we use in classes to exemplify things, such as the orchids, which have a very unique flower structure. We've got something like this, which has a different color for the leaves.
Okay.
Bromeliads,
So different
different morphology, some other flowers there. Yeah.
I love that.
Yeah, that's very nice. Very nice indeed.
In the wild, how does that grow? I'm sure they don't hang.
Probably hanging over rocks and
okay.
down ledges and stuff. Got a big fern here.
Yeah, this guy, what kind of staghorn?
Staghorn fern, that's right. Usually grows up in the trees as an epiphyte. A couple different ferns over here as well. Very nice. You can see the soft rabbit's feet in there.
Wow.
Rabbit's foot fern.
Huh.
Very, very quaint. Small there as well.
Being a vegetarian, I find the work that Dave is doing quite fascinating. When Ion WP returns, we'll go from labs and greenhouses to the great outdoors, where we'll experience the heights and depths of environmental science. Please stay with us. Welcome back to Ion WP. The biology department is more than just experiments taking place within its various laboratories. Important research is also being done in the field. Professor Lance Risley, for example, is engaged in a study of bat abundance, distribution, and species diversity, funded by the Federal Aviation Administration. His studies take he and his students into the field for hands-on investigation.
Got it, John?
His research involves the capture, measuring, tagging, and cataloging of various bat species in their native environments. Projects such as these are valuable to the students, the university, and the community at large. The students gain invaluable hands-on experience, the university prospers, and the community gets information to help solve problems and improve quality of life. Also getting his feet wet in the environmental waters of North Jersey is Professor Mike Sebetich. We had an opportunity to visit with Professor Sebetich and his class on a recent excursion to the Paquonic River.
Okay, our objective here today is to sample the Paquonic stream looking for invertebrates, mainly insects, and other invertebrates as water quality indicators. And so we have a group of teachers, 6 teachers from local schools who are learning the techniques for the first time. And we issued a small invertebrate chart that shows organisms that are sensitive, that are pollution tolerant, and those are in between. And we're going to attempt to see what kinds of invertebrates we can find here in the stream today and place in these 3 categories and try to estimate the water quality of the Paquonic River at this location. All right. Here's a beautiful stonefly. Stonefly, very sensitive to water.
Oh my gosh.
And so we're happy to find a stonefly with the 2 cerci at the end. And that means the water quality is fairly good, even though it's in an urban area. Well, we offer these activities because we want to teach our basic student who wants to become a biology major, plus we want to reach the local teachers. To educate the local teachers so that they can take the same techniques back to their schools and they're not afraid to do ecology. The problem in the past with doing ecology is the teachers didn't know what to do and how to do it. So we use very simple techniques, theory and ideas, which they can modify, use at their schools with their students in their local streams and their ponds. And so it's a good educational tool.
Yeah, my favorite part was getting out there and actually being able to pick things up and find them and see where they live and having the hands-on experience of catching all the invertebrates today.
Professor Sebetich's course exemplifies the William Paterson philosophy of live it, learn it, as well as our commitment to community outreach. Also working in the field with Dr. Sebetich is Dr. Richard Pardi, who takes a moment to tell us about a recent class expedition dealing with the dilemma of invasive species in an urban environment.
We will be taking the students out to look at what's called biocontrol of invasive species. In an urban environment like this, you get many, many species of plants and animals that don't belong here. They come in on people's feet, they come in on automobiles, they come in from planes, or they just crawl themselves in here somehow. There are a lot of invasive species here, and very often the invasive species will push the natural populations out. And there are a couple of invasive species right here, right now, right behind me, Canada geese being one of them. In order to restore these environments to their natural state, it's important to try to get those invasive species under control. So on Thursday, Dr. Vail will be taking them to another site nearby where we're actually going to do it. We're really going to go out and actually do some invasive species control and show these teachers how it can be done on one particular plant species.
What Dr. Vail's class will be doing is attempting to control an invasive plant known as loosestrife. They will do this by introducing into the environment a natural predator in the form of the Galarusella beetle that feeds exclusively on loosestrife.
You can see the pollinators going to town on these. Some native bees, which bumblebees, but they're also our European honeybee, which is a native encouraged invasive species, if you will. They're moving through the flowering season pretty quickly now. It looks like some of these spikes are almost done. This one's got maybe 4 inches to go. So that spike will expand a little bit more and the flower will kind of move up. One of the strategies for the plant is to have some flowers out all the time to keep those pollinators coming. So, you know, a single flower may only have a lifetime of 4 days, but that spike on that plant is going to be flowering for several weeks at least. And again, one of the things I want you to see right now, and we'll contrast this with where we've seen the biocontrol system established, no damage. So take a look at the leaves. Remember the photographs we looked at. These are untouched. One question I don't know the answer to is what protects these. We have lots, you know, not all herbivorous insects are specialists, and something would eat this if it could. Specialization as a general rule, on the surface of it, seems like a bad thing. Why would you want to be a beetle that only eats one kind of plant? And that's where the coevolutionary arms race comes in. In general, it's good to be able to eat a lot of things, but if it requires very special biochemistry to eat this, then you probably can't eat anything else. So. And my guess is that's like 5 years old. That's a guess. 3 to 5 years old. And they can get much larger, but you know, that might live at least another 5 years, and at 2.5 million seeds per plant, man, that's a lot of reproduction.
I hope that you've enjoyed this edition of our show, and I hope that you'll join us in the future as we discover more about William Paterson University through our Eye on WP.